Hi everyone, welcome to our tutorial, our short, quick crash course tutorial on basic immunology for M1s. I'm Joshua,
And I'm Akira.
and we'll be going through immunology for today. Okay, so very quickly, this slide gives you a framework of what you need to know in immunology. We'll be going through the specific details of these one by one during our tutorial today. You can categorize immunology into two segments: what happens in absence of the pathogen and what happens in the presence of a pathogen. So on the left-hand side, you note that in the absence of pathogen, you have firstly the anatomical organization of the immune system, where immune cells are compartmentalized differently in different places to serve different functions. And then your T cells and B cells also have a development and activation process that you will need to learn. In the presence of a pathogen, then your innate and adaptive immune response are kicked in with the help of important cytokines and receptors, and eventually you have the downregulation of the immune response. So first, we'll be talking about the anatomical organization of the immune system. You have two kinds of lymphoid organs in the body. The first is a primary, which are also known as a generative lymphoid organs because they generate the lymphocytes from stem cells. And the second is your secondary lymphoid organs, also known as a peripheral lymphoid organs. This is where antigens are presented to lymphocytes for activation of the adaptive immune response. So in the primary lymphoid organs, there are three main organs that you need to know: the fetal liver, bone marrow, and thymus. The fetal liver is only active in hematopoiesis, in the generation of lymphocytes during the fetal stage, which means when you're a fetus. Bone marrow is active in maturing B lymphocytes. The thymus is active in maturing T lymphocytes. You do note that actually for T cells they derive from hematopoietic stem cells in the bone marrow, and then these precursors migrate into the thymus for further development. For secondary lymphoid organs, you have your spleen, lymph node, and your cutaneous lymphoid tissue. Important difference we need to know is that the spleen filters blood, therefore it picks out the blood-borne antigens for activation of immune response. The lymph node, because it drains lymphatic fluid from the epithelial tissue, therefore it presents antigens from epithelial tissue to lymphocytes in the lymph node. So this is an important difference. The spleen filters blood-borne antigens, the lymph node filters lymphatic fluids from epithelial tissues, therefore antigens presenting from there. However, in the body, there are many surfaces that are exposed to the environment and therefore can be common sites of infection. These include your GI tract, your mouth, and your skin. So therefore, you have things like Peyer's patches and tonsillar lymphoid tissue in some of these areas, so that your body can mount a fast response to any antigen that is presented within that area. It's important to note that they do not respond to commensal flora. This means that for example, the Peyer's patches in the GI tract do not respond to bacteria that are part of the normal microbiome in the GI tract. Importantly, on the bottom left-hand corner, you must recall the compartmentalization of lymphocytes within each of these lymphoid organs. Remember that lymphoid organs are organized into follicles, and within the follicles, you'll find your B cells and your follicular dendritic cells. In the paracortical regions, you'll find your T cells, which will present the antigens to the B cells, and then you will also have the germinal center within the follicle, which is where B cells differentiate into plasma cells. Please review histology for this.
Alright, so now I'll just be talking about T cell development, which is part of hematopoiesis. Okay, so very quickly, this is just an overview of the whole development and activation process. And the main point that you're trying to make in this slide is to make a differentiation between immature/mature cells and naive/effector cells. Okay, so it's very easy to get confused between like mature and naive, that kind of things. Okay, so just remember, immature T cells is before it develops. Like this happens within the thymus/bone marrow for T cell and B cell respectively. Okay, so maturation is part of the development process. So after you have your mature T cells/B cells, which will be in your primary generative lymphoid organs, and then it's a naive T cell/B cell, and then it will be activated due to the presence of due to like recognition of antigens. Okay, and these activation processes will lead it to be differentiate into an effector T cell or effector B cell. And on the right hand side, you can see all the different subclasses of all your effector T cells or your effector antibodies, and this will elaborate more later. Alright, okay, so now we'll be talking about the development of the T cells. Okay, so in T cell development, there are two main processes that you need to know about, and this is VDJ recombination, that's one, and positive and negative selection, that is the second one. Okay, so firstly, the key point is that you need to know that these two processes, the purpose is to produce a wide variety of receptors that can recognize a wide variety of antigens. Okay, so let's start with the first one, which is VDJ recombination. Okay, so the purpose of so the aim is to diversify the T cell repertoire, and in a nutshell, the two main things that you need to know is that there's combinatorial diversity and junctional diversity. Okay, so for combinatorial diversity, how you can think of it is that, for example, let's say I'm going to make a IG heavy chain, okay, and then in the gene segment, there are like, for example, 50 V segments and maybe 20 D segments and maybe 5 J segments. So you can really see that there's like 50 times 20 times 5 different possible combinations that you can produce. And that's quite a huge number already. But it's still not enough in the context of immunity. So we have junctional diversity. And this is where, in between the V, D, and J segments, there'll be random addition of nucleotides. And this is random. So it means that there's really a huge repertoire of receptors that are being coded for. But the thing is that now that we have produced such a wide range of receptors, then we need to select for receptors that, firstly, they're actually useful, and secondly, we have to select against receptors that will recognize our self antigens. They are autoreactive. Yeah. Okay, and this is where part B comes in. So that's positive and negative selection. Positive selection is in overview, positive selection is where we select for receptors that can recognize MHC, and negative selection is where we select against those that are autoreactive. Okay, so we refer to the picture below. So you see that at the start, from stem cell, it will become a double negative, CD4 minus CD8 minus T cell, which means it doesn't express both CD4 and CD8. And then you just develop more and more until it becomes double positive. So at this point, we call it an immature T cell, and it has both CD4 and CD8. Okay, so what happens now is that this happens in the thymus, and thymus has cells that have MHC, class 1 and class 2, with a self-antigen presented on it. Okay, so the T cell will go around like recognizing all these antigens. And then after this, there'll be 4 possible outcomes. Okay, the 4 possible outcomes are listed out here, but let me just read it to you. So positive selection is when we have weak binding of the receptor to either antigen processed on a class 1 MHC or a class 2 MHC. If the receptor binds to the class 1 MHC, then the CD8 plus receptor will remain, while the CD4 plus the CD4 receptor will disappear, so it becomes CD8 plus only. And as for class 2 MHC, if the receptor binds to the class 2 MHC, then the CD4 receptor will remain and the CD8 one will regress. Okay. Then on the other hand, if it does not bind to any MHC antigen complex, then the cell will die. And at the same time, if it reacts too strongly to a self-antigen presenter of MHC for either class 1 or class 2, it will also die. And this is called negative selection. Yeah.
Okay, now we will run through B cell activation. So just to recall the earlier slides, activation is the process by which a naive B cell becomes an effector B cell. So the development of B cell is not within syllabus, so it does not need to be learned. But you do need to know the processes by which B cells are activated. So looking at this slide, remember that we are now discussing mature B cells that are naive and not activated, and this is the process by which it is activated. 2 main processes you need to know. The first is somatic hypermutation, and it is a somatic hypermutation that allows for the second process, which is affinity maturation. So let me direct your attention to the picture on the right. In the dark zone of the germinal center, you have centroblasts. So these centroblasts are your mature naive B cells, and what happens is that upon activation, they will polymutations will be introduced to the antibody receptors that are expressed on these proliferating B cells, and these antibodies will all have different specificity to the antigen as a result of these mutations. This allows for the centroblasts to undergo affinity maturation. The centroblasts will move from the dark zone into the light zone, where they will be renamed as a central site, and it is over here where the central sites compete against each other for binding to antigens that are presented on the follicular dendritic cells. These antigens presented on the follicular dendritic cells are also known as icosomes. Therefore, the outcompeting of between central sites allows for a situation where central sites with high affinity receptors to these icosomes will outcompete those with low affinity receptors. And because there is only a limited number of icosomes available, because there's a limited number of antigen, as the number of antigens still available decreases, you will have a situation where a large number of B cell central sites are competing for a very small number of icosomes that are remaining. Therefore, only the strongest B cells can outcompete and bind to the icosome, and therefore get the signal transduction events which allow it to leave the germinal center and mature into either an effector memory B cell or an effector plasma cell, which then goes on to produce various antibodies such as IgG, IgM, IgA, and others. Okay, now we will be covering what happens in the presence of a pathogen when an infection is about to occur. Akira will first give us an overview.
Okay, so this picture here is an overview of what happens, how our immune system responds to a pathogen. Okay, so just a roadmap. Okay, so first we have innate immunity on the left and it's in purple, and then we have adaptive immunity on the right in red, and below that we have the cell-mediated portion and the humoral portion. So the cell-mediated one is the one in pink, and the humoral one which is in the one in orange. Yeah, so just remember that the cell-mediated one is T cell and humoral one is B cell. Okay, so let's head over to first before that let's discuss on like the differences between the innate immunity and the adaptive immunity. Okay, so two main differences. Firstly, the innate immunity is a very general response, while adaptive immunity is very specific to a specific antigen. Innate immunity, upon repeated encounter of the same antigen, they will always produce the same response. Whereas for adaptive immunity, there will be stronger responses on a repeated encounter of the same antigen. So let's head over to the innate immunity portion. So it's in purple on the left. So under innate immunity, there are actually two main reactions that you need to know, and those are firstly inflammation and secondly antiviral response. Alright, so for inflammation, the mediators are macrophages and neutrophils. Okay, and the details you all should just read your lecture notes. But yeah, then antiviral, the main mediator is natural killer cells. Alright, okay, then as we move on to the adaptive immunity, right, there's a link, and that link is actually the antigen presentation, which is done by dendritic cells through MHC2. So what happens is that dendritic cells will capture the antigens from the peripheral tissue and then move to the lymph nodes, and then you present it to your T cells through the MHC class 2. Alright. Another form of antigen presentation is also on MHC1, and it's actually on the nucleated infected cells themselves. So what happens is that the CD8 plus cytotoxic T cells will come and recognize these cells and then kill the cells. Yeah, so that's the bridge between innate immunity and adaptive immunity. So now when we move into the adaptive immunity part, so as I mentioned, there's cell-mediated and humoral. Okay? So cell-mediated, for now, I'll just say that there's two types of T cells, CD4 plus, T helper cells, CD8 plus, T cytotoxic cells. And I also say that under CD4 plus, there's actually a different types of effector cells, like Th1, Th2, Th17. There's actually a T follicular helper and a T regulator also. Alright. Then for the humoral part, we have a T-dependent reaction and a T-independent reaction. So just remember that the main one is T-dependent, but the T-independent one is activated when a B cell encounters, for example, a polysaccharide or a lipid antigen. And the T-independent one is a very weak reaction and involves only IgM. Yeah. So for the T-dependent reaction, we have actually a whole different range of Igs that are involved. So for example, IgM, IgG, IgE, IgA. And this occurs because of isotype switching, which is actually induced by your T helper cells. Yeah. So that was an overview of the immunity, but now we are left with the complement system. And actually this complement system is a combination of both the innate immunity and the adaptive immunity. Alright. So as you might have learned, there are three pathways that lead to the complement system and three things that result from the complement system. Alright. So I'll just explain this shortly here because we won't be talking about this later. Alright. So the first thing is a classical pathway, and this is antibody mediated. So I've written here is mediated by IgM or IgG. And what happens is that IgM and IgG will bind to your microbe surface, and that would cause a cascade to occur through C1. By the way, the complement system uses proteins C1 to C9. Okay. So what happens is that when IgM or IgG binds to the microbe body, then it will recruit C1 to come over, and once C1 comes over, then there's a whole cascade which eventually results in C1, C2, C3, C4 binding together and then causing the conversion of C5 to C5a. And then another cascade occurs, and then you form a membrane attack complex which drills a hole basically into your cell, bacterial cell, and cause osmotic lysis. Alright, so that's the classical pathway. For alternative pathway, what happens is that your C3 fragments bind directly to the microbe, and then a similar thing happens, C5 will convert to C5a, and then the whole membrane attack complex happens. And the lactin pathway is essentially the same as the classical pathway, except that there's this molecule called lactin that binds directly to your cell or your bacterial cell and acts like a C1 protein, and then everything's the same as the classical pathway already. Alright, so that was the pathways that lead to the complement system. Now you have to think about how it goes on from there, the three reactions, and those are opsonization, cell lysis, inflammation. Alright, so very briefly, opsonization, basically, I think it's C3, D, something like that. You coat the bacterial cell wall and make it more favorable for macrophages to like phagocytate. So it's like, think about it as it becoming more yummy for the macrophages to eat. Yeah, cell lysis is by the membrane attack complex, and inflammation is actually by the production of chemokines, which are C3A and C5A. So these would recruit your macrophages, your neutrophils, and everything to come over to your site of infection. Yep, so that was the complement system.
Okay, so just to reinforce, the classical pathway is considered like a part of the adaptive immunity because it is facilitated by IgM and IgG antibodies, whereas the alternative and lectin pathways are means by which the innate immunity can start and initiate the complement cascade to produce those three effects. This slide shows you a very nice overview of all the activities and the mediators that are involved in the entire immune response in response to pathogen intrusion. So we'll start from the top. Pathogen intrusion always begins when there's a break in physical barriers, and the main concept is that whether it is our skin, our tears, our gut, there's always some kind of a flushing activity that takes place so that we can keep washing away any pathogens that may be stuck onto these mucosal or epithelial surfaces. And therefore, an absence of these flushing mechanisms such as peristalsis in the gut or tear secretion in the eye or the layers of keratin in the skin can then cause a break in physical barriers and lead to pathogen intrusion. Again, big picture, the left-hand side in purple is the innate immune response. The black color refers to the link between the innate and adaptive immune response, whereas the diagrams in red represent the adaptive immune response. So with the pathogen intrusion, let us start with the innate immunity. Recall that this is a much faster acting immune response, whereas adaptive takes like hours, days to take effect because you need to have an antigen presenting before it can be activated. Okay, so every pathogen can stimulate what we call DAMPs or PAMPs. So DAMPs is danger-associated molecular patterns, and PAMPs is pathogen-associated molecular patterns. And so, what happens when there's a pathogen intrusion is that certain surfaces or polysaccharides, glycoproteins that are present on the surface of the pathogen may be detected by receptors in the body as a pathogen-associated molecular pattern (PAM). A pathogen intrusion can also lead to cell damage of the cells that is infecting, and these cells, when they undergo lysis or apoptosis, they release their intracellular contents, and these can be recognized as danger-associated molecular patterns. These DAMPs and PAMPs are then recognized by two kinds of receptors, which may be intracellular or extracellular. They have the NOD-like receptors and the TOE-like receptors, of which the TOE-like receptors was covered in your lecture notes. These receptors are present on the surface of your resident immune cells, such as your macrophages, your natural killer cells, and your dendritic cells. And this is what causes their activation. So, upon activation, what happens is that these resident immune cells will release cytokines, and these cytokines will facilitate inflammation, which will be discussed later on. The resident immune cells will also then go on mostly to phagocytose or to induce apoptosis in these infected cells. So the natural killer cells provides the antiviral function, as Akira covered earlier on, by inducing apoptosis in virally infected cells because viruses tend to be intracellular and to hijack the host cell's machinery. On the other hand, other microbes can be phagocytosed by macrophages and dendritic cells. Macrophages can either immediately kill these microbes within their phagolysosomes, or they may function like dendritic cells as antigen-presenting cells, migrating to the lymph nodes via the high endothelial venules and then presenting them to the adaptive immune system. Okay, now let's zoom out again and look at complement cascade activation, which is a part of the innate immune response in this diagram. So the pathogen intrusion can immediately activate your complement cascade, which results in three functions as Akira discussed earlier on. Membrane attack complex punches a hole to induce osmotic lysis in the cell. Acute inflammation is mediated by cytokines TNF, interleukin 1, 6, and 12, and this will result in the ultimate recruitment of more leukocytes that are non-resident in the area from the blood, so that there can be a well-mounted innate immune response. Interferon gamma is an important cytokine that stimulates macrophages to spur its phagocytic function, and also its release of reactive oxygen species and nitric oxide, which are like your bactericidal agents that kill microbes having been ingested by macrophages. Now let us move on to the black section of this diagram. Okay, hang on. Okay, actually before that, let us talk about acute inflammation. Okay, now let us discuss acute inflammation first. So acute inflammation in detail, as I said earlier, was mediated by these cytokines over here, which are the signaling molecules of the immune system. There are three stages of inflammation and three processes that allow leukocytes to transmigrate from the blood into the site of infection. And this you should be familiar with.
Okay, so now we move on to the black part, which is your antigen presentation. It's the link between your innate and your adaptive immune system. Okay, so basically, as you know, there are two classes of MHC: MHC class 1 and MHC class 2. Alright, and in MHC class 1, we are dealing mainly with intracellular antigens, while for class 2, we are dealing mainly with extracellular antigens. Alright, and another difference is that MHC class 1 is present on all nucleated cells, while MHC class 2 is only present on antigen presenting cells such as the dendritic cells. Alright, okay, so the process for antigen presentation using MHC class 1 is that what happens is that, for example, let's say we have a virus which infects a cell, alright, and then inside the cell you have viral proteins that are being produced because the DNA is inside the cell and it's producing proteins, alright, and then the viral proteins will be ubiquitinated, and then you go through the proteasome and you break down into peptide fragments, and then at the same time you have your MHC class 1 that is being synthesized in your ERs. So what happens is that these peptide fragments and first the peptide fragments have to go from the cytosolic compartment into your ER, right? So you have to go through this TAP molecule to go in. It's a transporter, and once it goes into the ER, your MHC class 1 and your peptide fragments can meet together. Okay, so there's this special thing about MHC class 1 and MHC is different from MHC class 2 in that this invariant chain is not being produced. I'll talk more about the invariant chain later, but just remember in MHC class 1 you don't have an invariant chain that is stuck with the MHC complex. Alright? Okay, so back to your MHC class 1, what happens is that in the ER, the MHC class 1 and your peptide fragments, which are from the viral protein, they'll come into contact, and then your peptide fragments will get loaded onto the MHC, and then you get processed in the Golgi apparatus, and then you move to the cell surface to get expressed on the cell surface. So that's how your antigen, your intracellular antigens, will be processed on your cell surface for recognition by your T cell receptors, your CD8 cytotoxic T cells. Alright, so that's class 1. So now I'll be talking about the MHC class 2 extracellular antigen presentation. Alright, so now we start from the expression of MHC class 2. So if you remember, I mentioned that in MHC class 1, we do not have an invariant chain being expressed as well. So now the reason why MHC class 2 has an invariant chain is so that self-antigens that are floating around in your ER won't go and bind to your MHC class 2. So the invariant chain is stuck onto your peptide binding cleft for your MHC2, and then you block your self-antigens. Okay, so after it's expressed, then the MHC class 2 along with the invariant chain will be shunted off, and then what happens is that now you have your virus, your bacteria, your fungi all endocytosed, right, by your antigen presenting cells, and then you form a phagolysosome, and then in the phagolysosome, your bacterial protein, your virus protein, fungi protein, will all be broken down into short peptides. Then these are your antigenic peptides. So the phagolysosome will meet together with with your MHC class 2 with your invariant chain. And what happens is that the proteasome that was in the phagolysosome, now it will go and break up the invariant chain to form your clip segment. Okay, and your clip segment is what allows your antigens to go and replace the clip, so that now your antigenic peptides are being bound to your peptide binding cleft on your MHC class 2. And when that happens, then your MHC class 2 can move out to your cell surface to be expressed on the cell surface, and then your CD4 can come and recognize your MHC class 2 complex.
Okay, so now we have discussed how the innate immune system links to the adaptive immune system through the processing of the major histocompatibility complex 1 and 2, by which to recall, MHC1 presents intracellular peptide fragments, MHC2 presents extracellular antigenic peptide fragments. So now we'll be talking about this B7 CTLA4 and CD28 interactions. So what goes on over here, as you recall, you actually have two kinds of T cells. You have your cytotoxic T cell and your helper T cell. Your cytotoxic T cell carries a CD8 receptor and a CD28 receptor. Your naive helper T cell carries a CD4 and a CD28 receptor. And these receptors are what interact with the MHC2 and B7 ligands present on your antigen-presenting cells during the activation of the T cell. How does this happen? So there are three signals that are required for the activation of T cells. The first is that the T cell receptor, whether it's CD4 or CD8, must bind to the MHC complex by which the antigenic peptide is being presented. The second signal for activation of T cell is that it requires what we call co-stimulation, and this is offered by the CD28B7 interactions. So going back over here, you can see that you can remember that on the dendritic cells or your antigen presenting cells, you have B7 molecules which are ligands that are expressed on the antigen presenting cell only when a pathogen has been detected and processed within the antigen presenting cell. So let's say I'm the dendritic cell. I have processed my extracellular pathogen, so my MHC2 is ready. So at the same time, when I express my MHC2, I will also express my B7 molecule. And the B7 will interact with CD28, your MHC2 will be recognized by CD4. So let's talk about the CD28-B7 interaction. What happens over here is that the binding between CD28 and B7 will result in a signal transduction event within the T cell that encourages its activation. At the same time, when it is activated, you will then have a massive release of interleukin-2, and interleukin-2 receptors expression increases on the activated T cells as well. This allows for an autocrine effect that facilitates the clonal expansion of your T cell. Why this is important is because, as you remember, we have the development process has developed a wide repertoire of T cell receptors on T cells, but there's only one type of receptor on one T cell. So when that receptor finds an antigen that is specific for and it can bind to, that particular T cell needs to self-replicate to produce many more T cells that can respond to the same antigen with the same specificity. This is why clonal expansion is required. And it is this release of interleukin-2 and the increased expression of interleukin-2 receptors on the activated T cell that facilitates this clonal expansion, because interleukin-2 is an important co-stimulator that proliferates the T cell. Yes. At the same time, during activation, you also have the increased expression of what we call CTLA4. So going back over here, in green over here, this is CTLA4. So just when interleukin-2 and its receptors are being expressed by activated T cells, the activated T cells also express CTLA4. This CTLA4 will compete with CD28 for binding to the B7 molecule, which is what is demonstrated in the picture over here. Because CTLA4 has a much higher avidity to binding to B7 molecules on antigen-presenting cells, therefore the CTLA4 will outcompete the CD28 for binding to the antigen-presenting cells B7 molecules. What happens then is that the CTLA4 facilitates an inhibitory signal that prevents an overactivation of the T cell, so that our immune response is always well controlled. Because remember, if the immune response becomes overwhelming, what will happen is that you will develop autoimmunity, where basically your immune cells start attacking the whole cells. Okay, I hope the explanation was clear. Now, I will be discussing the activation of how the effector T cells. Okay, I hope the explanation was clear. Now, we'll be moving on to discuss what happens when a T cell is activated. So activated T cells can manifest as either Th1, Th2, or Th17 T helper cells. These are the main classes of T helper cells. You also have your T follicular helper cells and your T regulatory cells, but this we can talk about it later. The Th1 cell responds to intracellular bacteria and parasites. And how it does that is by increasing secretions of interferon gamma. Remember that interferon gamma facilitates phagocytosis by macrophages. So this will help M1 macrophages clear the phagocytosed pathogen by stimulating more reactive oxygen species and more nitric oxide to kill off all the microbes that he has ingested. Because if not, it's just going to be stuck in the macrophage, and the macrophage will become an infected cell. Next, we have the Th2 helper cells. So the Th2 helper cells, their main activity is in responding to helminths. Helminths are things like your roundworms, which often infect the GI tract. So how this happens is that Th2 releases cytokine interleukin-4 that contributes to the helminth response. M2 macrophages are macrophages which facilitate repair of epithelium, healing and repair of damaged tissue. And so what this does is that it repairs the GI tract, so it prevents all the helminths and worms which have infested the GI tract from entering the bloodstream. At the same time, interleukin-4 also increases peristalsis, so it can sort of flush out all the helminths that are in the GI tract, and it also induces mast cell degranulation, because the chemicals such as histamine within mast cells are toxic to helminths. Th17 helper cells facilitate your extracellular pathogen response. What they do is by amplifying the recruitment of leukocytes via inflammation, as you recall, which was a part of the innate immune response, and they also increase the barrier integrity by beefing up the physical barriers that prevent pathogen intrusion in the first place. These are mediated by interleukins 22 and 17. Okay, here is just a summary slide that shows us all the important cytokines that you will need to know for the purpose of your exams. Okay, so now let me just say a few words on how the adaptive immune system returns to homeostasis so that it is not constantly activated. This is facilitated mainly by the interaction between PD-1 and B7 in green, and also by your T regulatory cells. Let me start by talking about T regulatory cells. As implied by the name, T regulatory cells regulate the immune response. They do this by secreting cytokines like interleukin 10 and TGF-beta, which inhibit the further activation of leukocytes, your T leukocytes, your B leukocytes, and other stuff. That's for T regulatory cells. And the interesting thing to note over here is that, remember how I said interleukin 2 was the main cytokine that facilitated the clonal expansion of T cells? So as much as interleukin 2 facilitates the activation and expansion of T cells in response to the pathogen, interleukin 2 also helps to maintain T regulatory cells to prevent them from apoptosis, from killing themselves. So this helps to provide a homeostatic balance to the immune system. At the same time, activated T cells will also express PD-1, which will respond to PD-1 ligands on the antigen-presenting cells that shut down the activation cascade within the signal transduction events in the T cell. Okay, so now just a very quick word on the production of antibodies IgA, IgM, IgG, and IgE by your plasma cells. So if you recall, your B cells are activated by somatic hypermutation and affinity maturation, and now they become plasma cells. Most become plasma cells, and they need to secrete antibodies. So they will secrete either IgM, IgG, IgE, or IgA antibodies.
Okay, so yeah, so the purposes of the different IGs, right? Okay, so firstly, I'll talk about easy one. So IgA, okay, IgA is secreted at your mucosal lymphatic tissue, right? And then it confers like a sort of like a protection at your mucosal sites. So what happens is that, for example, when your pathogen comes in through your respiratory tract or your GI tract, your IgA will go there first and then go and neutralize all your microbes there, so that they won't even have a chance to enter your systemic your system. So that's your IgA. Okay, IgE is important for two things: your allergy and your helminths. Okay, allergy is not very relevant for you guys, but just remember IgE, helminth, and where did we hear helminth before? We heard it for Th2, right? So Th2, mast cell, IgE. These are the common things that you have to know about for helminths. Okay, IgG is your most potent antibody. Okay, it's for your opsonization and for your complement activation. Yeah. Okay. So what happens is that I mentioned before that without your T cell helping to activate your B cell, right? Your plasma cells can only secrete IgM, and IgM is actually a very minimal basal level kind of reaction. So there's your isotype switching to your IgG, IgE, and IgA. Yeah, and just now I mentioned all your effectors already. Okay, IgD, right? IgD is a special one. It doesn't actually get released from your plasma cell. It actually just stuck to your cell membrane. So it's sort of like a cell receptor kind of thing. Yeah. So your main ones are IgM, IgG, IgE, and IgA. Yeah. So okay, let me just recap the whole thing. Okay. So upon activation, IgM isotype switching to IgG, IgE, or IgA, depending on the requirements that your body needs. Okay. Let's say your body encounters a helminth, then your isotype switching will mainly be to IgE. Okay. Let's say your body encounters like for example bacteria in the blood, then your main one will be IgG. Let's say your mucosal lymphatic tissue encounters microbes in your GI system, then the main one will be IgA and the GI system. Yep. Okay.
Okay, I think that brings us to the end of our short tutorial over here. We hope it has been helpful. So just remember, this is everything that you need to know. If you have any questions, feel free to find Akira or myself via the ACAD-D helpline.